Okay, I think as we wait for some more people to join, welcome everyone to the fourth edition of Tech-novation. My name is Ravi Baid and I'm the co-founder for BIS Research. And along with me are two really expert folks who have done more than decade plus work in healthcare and research around cancer. Gabor, who is from Budapest, Hungary, welcome. Uh, Wahid, who is from New Delhi, from BIS Research, and is a vice president in research for BIS Research. Uh, and once I get a green signal, we will start our very, very interesting session, I think, which we intend to have. We intend to learn about this whole new technique to look at how cancer can be treated using, deuterium depleted water. So that before I get into the conversation and discussion, let me give you a quick introduction about what BIS research is. Some of you already know because you're coming from our LinkedIn page, but nevertheless, very quickly, BIS research basically has been in business for almost a decade now. We track deep tech and we track innovation within certain deep tech impacted markets. And we provide market intelligence to our customers, which are large Fortune 500 companies, startups, and they're spread across the globe. So that is what we as a company are doing. Uh, the market for healthcare is nearly a $10 trillion market and constitutes almost 10% of the global GDP, and it is growing at a rate of 9% annually. That's the kind of size of the market, and so healthcare for us as a sector is very, very important, and we track it. And within healthcare, we look at domains like oncology, gene and cell therapy, precision diagnostics, immunotherapy, and new metabolic interventions such as deuterium depletion. Okay. Our research looks at things which are in the lab, which are new, and which are going to get into the market, and we help companies navigate that entire ecosystem. As a company, what sets us apart is the fact that we've got our own proprietary research methodology. We have a pool of experts which are global. We use the knowledge which is acquired through a lot of primary intervention, and of course, today as a company, we have adopted AI like anybody else, so that we can do things at a faster rate and more, in a more speedy manner, right? So today's session, why is it, how does it matter? I think deuterium depletion is a promising breakthrough in the metabolic cancer therapy area, and it's offering a new frontier in precision medicine and treatment. So we will take today's session in a very interactive way. Uh, Let me get you to hear from our experts. So I will. Before I hand over, let me introduce uh, our experts today. We've got Geber Somlai, who is a PhD and is the founder and longtime CEO of Budapest-based HYD LLC. They specialize in cancer research and drug development, a very pioneering biotech focused firm which works in the deuterium depletion therapeutics space. Uh, I was speaking to Geber earlier, and I think he and his entire family is very passionate about the subject. So we hope to hear a lot of stories about your journey, through the session. Thank you for joining, Geber, today. Uh, then we have Wahid, Abdul Wahid, who is the Associate Vice President here at BIS Research. He's very, very passionate about healthcare. He spent nearly 13 years, in terms of researching the impact of healthcare on businesses. And, Wahid lives in Delhi with his twin 4-year-old boy and girl, and that's where he spent more, some of his time, what he can get out of after doing the research work which he does. So, without any ado, let me invite Wahid to lay out the landscape, of the topic today, which we are going to look at, and then we'll have a Q&A asking Wahid to then interact with Gabriel. Okay, over to you, Wahid.
And welcome, Gabor. And good morning, good evening, and everyone, depending on your time zone. So let me start with, I'll give you just background of the topic that we are dealing with today, especially the cancer. And then we'll jump with discussion on the deuterium-depleted water and its application and different aspect with Gabor. So let me share my screen for a second.
Yeah, sure. Thank you for having me. So, should I summarize it briefly?
Sure.
Yeah. So I should go back more than 50 years when a Hungarian Nobel laureate, Albert Szent-Györgyi, who discovered the vitamin C, visited Hungary. And in his interview, he said, if you want to solve the cancer problem, you have to go sub-molecular level. I was very young, teenager at that time. And Albert Szent-Györgyi, he was a very clever Hungarian scientist because his way of thinking was that how is it possible that we can and the cells can respond so fast for all the challenges. Meantime, the proteins are so rigid, they are so big. So he raised the question whether is it possible that the life is organized by that molecules, big proteins, which the synthesizing the proteins takes long time, the induction of the genes takes long time. So that word submolecular, that was the very first point which which was very important in that long journey. So I kept it in my mind that submolecular, and Alberti suggested that particle should be that negatively charged electron. And one day, that was in 1976, May 21st, I thought, okay, maybe that proton should be that submolecular particle, the positively charged hydrogen ion. So I make a note and I wrote down that the hydrogen ion responsible for the cancer. And that was in 1976. And then I went to the university, and 4 years later at the university, the other idea came to my mind that there is a heavy hydrogen, it is called deuterium. And at that time, that was in 1980, I was sure that somehow the deuterium and the hydrogen together regulate the cell growth and responsible for the tumor development. I graduated in 1982. Then I continued my work. That was a DNA work. I isolated genes and identified genes. I had a fellowship holder for the Hungarian Academy of Sciences. I won my PhD. I worked in Germany. I went to the United States. When I came back from the United States, I started to look for a job which is related to the cancer, because that was my original plan to do research on the cancer field. So in 1990, I started to work in the Hungarian National Institute of Oncology. And my question was to investigate whether this deuterium has any role in living organisms. The people should know that the deuterium was discovered almost 100 years ago. And it was clear that due to the 100% mass difference between the hydrogen and deuterium, that these two hydrogen behave differently in chemical reaction. It is called isotopic effect. And for 60 years, the heavy water was tested in different biological systems, and it was clear that it's for a living organism, it's not the same having a deuterium or hydrogen, because you can kill a dog with a heavy water over 35%. So it was clear that even both is hydrogen, in the chemical aspect, we can talk about two different elements. And the other disturbing thing was, I guess, in the last 100 years, that the ratio between the hydrogen and deuterium is about 6,600 to 1. So if we take 1 million hydrogen atom, there will be only 150 deuterium atom out of 1 million hydrogen atoms. So this is the reason we say that the deuterium level is about 150 ppm parts per million. And I suppose that the scientists, because that ratio, 6,600 to 1, believe that, okay, let's ignore the deuterium. It is so rare. Why should we think about deuterium? And that was a question what I addressed. How can we ignore the deuterium? If we express the deuterium level in millimole, it turns out that deuterium level in our body is 6 times higher than calcium and 10 times higher than magnesium. So this is the question I address. What will happen if we reduce the deuterium level, and what kind of consequence we can detect in the biological system? So in the very first paper was published in 1993, and in that paper we proved that the lower D level reduced the cell growth in vitro, and when we replaced the drinking water of the mice which had been transplanted with breast cancer, we could save the life of the mice, which is clear indication that first, the deuterium hydrogen ratio is critical and has a big impact on the cell growth. The second, the deuterium depletion has an anti-cancer effect, because in these mice, the cancer disappeared when the mice started consuming DW. So when we received that type of results, I quit at the institute and we established the company, the HYD, and I promised my partners that we will register the best cancer drugs of the world. That was more than 30 years ago, and we are still working on that to convince the world and to get the approval and to take part due to depletion of the oncotherapy.
Right.
And one of the questions always whether we know the mechanism. And we say that yes, we already know the mechanism because we have invested so many time and fun to figure out what's going on. So everybody every day synthesize about 0.34, zero point 0.4 liter deuterium depleted metabolic water. So when we are eating the food and we burning it, carbon dioxide and metabolic water, that metabolic water is deuterium depleted. And how depleted, it depends on what type of food we eat. The experiment we done was very simple. We went to the supermarket, we bought different kind of food, and then we measured the deuterium level of the dry components of the food. And it turned out that from the carbohydrate, the cell synthesized metabolic water with 150 ppm, so like normal deconcentration. But when we burned the fat oil, that was 118 ppm or 109 ppm. So that means the more fat we eat, the lower will be the deconcentration of the metabolic water.
Yeah.
And why is it important? Because the brake system in the cells is in the mitochondria, and the mitochondria producing the deuterium depleted metabolic water. And the brake is very important because when a growth hormone binds to the membrane and trigger the cell growth, that will stimulate the so-called sodium hydrogen exchanger, which means that the cells pick up one sodium and transport one hydrogen. And in our very first paper, we recommended that when the sodium hydrogen transport is activated, that will be separately transfer only the hydrogen, which means the activation of the sodium hydrogen antiport will cause an increase in the DH ratio.
Yeah.
And we said this is the signal for the cell, which are when the DH ratio within the cell reaches threshold, that will turn on hundreds of genes, which expression is essential to manage the G1 phase to the S phase, doubling the DNA, and then close the cell cycle. So in a healthy cells, the membrane transport can increase the DH ratio, but in a healthy cells, the mitochondria produce deuterium depleted water, so they are imbalanced.
Yeah.
And this is another story which is well known for almost 100 years, that the only common feature in each cancer is the damaged mitochondria. So in a cancer cells, the mitochondria is not able to produce deuterium depleted metabolic water. So this is the reason when there is the activation of the growth hormone stimulate the sodium hydrogen antiport, that much easily can lift up the DH ratio to the threshold, and then the cell will divide and will multiply. So that's it.
Right.
And so the very important message, when we modify the DH ratio, that will simultaneously modify all the enzymes, because will be a new equilibrium for the DH ratio. So when the DH ratio is increasing, then in each molecules, in the proteins and all the molecules which has a hydrogen in an exchangeable position, then more deuterium will bind in that protein and other molecules because we increase the DH ratio. So that send a message to all the molecules. And this is how the cells can be organized, because it is so complex. But even the complexity, it is well organized and can manage all the challenges. So
Right.
this is a basic mechanism.
Right.
So
So, Gabor, we understand it's a very complex mechanism in any cancer cases, right? As you mentioned about the concentration of DDW, deuterium depleted water, is it constant or have a threshold value same for every cancer, or it depends on cancer to cancer subtype? For example, the DDW concentration be effective for lung cancer could be different from breast cancer, or is it same?
yeah, so in the last 30 years, we have followed thousands of cancer patients with roughly 60, 70 different cancer types. And it is clear that the response rate is different depending on the cancer.
Okay.
The good news that those cancer types which represent roughly 50% of the cancer population, lung
Right.
cancer, breast cancer, colon cancer, prostate cancer, they responding quite well. We can get a 3 to 10-fold increase in median survival time with that type of cancers. But the glioblastoma, which is one type of brain cancer, the pancreatic cancer, they are so aggressive that even we are, let's say, we can get a 2, 3-fold increase in median survival time. We see that we need lots of research to continue to increase the efficacy in that very aggressive type of cancer. And the final data and the final results depends on three critical steps. We just recently we published a paper. We evaluated 2,649 cancer patients, and we evaluated the survival.
Right.
And our conclusion is that the date is critical between, so when start the patient consuming DW comparing to the date of the diagnosis. If the patients within 9 months start drinking DW, the chance for a long survival or to be in remission is great. The patients should have a 3 to 4 months life expectancy when they start to consume DW. So we cannot say that independent of the staging, we can turn back the process. And the other very important factor, the patient should drink at least DW for 3, 4 months.
Yeah.
So if they start within 9 months after the diagnosis, they can live longer than 4 months when they start, and they drink it longer than 3, 4 months. Then the median survival time was 11.6 years, which is a 5-fold increase comparing to the cancer population worldwide. And that conclusion is that the integration of deuterium depletion to the existing therapy can reduce the deaths caused by cancer by 80%, which is a very big difference
Right.
comparing to the data what we have today.
Right, right. And Gabor, as you rightly pointed, I mean, the breast cancer and lung cancer itself, I mean, the huge subset of entire cancer indication. And WHO predicts in next 3 decades, so by 2050, we'll spend almost $6 trillion just managing breast and lung cancer. So it's a huge, huge, huge expenditure for us, and there could be a large market, of course, for any therapeutic product that has subsequent impact on it. So, Gabor, do we have any longitudinal study that done on those cancers, especially on lung cancer or breast cancer, where we are looking at long-term outcomes for DDW?
Yeah, we have published papers with 3 papers, if I remember, with lung cancer.
Hmm.
Uh, two of them was the evaluation of 126. The other was over 200 cancer patients. But for example, one of our paper was published about patients who had already a brain metastasis from the lung cancer.
Hmm.
And it was very interesting that one of the lady was uh, uh, expected 2, 3 months survival because the non-small small cell lung cancer metastasis in the brain, and she started consuming DW, and she was fine for over 12 years. So instead of 3 months survival, she had 12 years survival, and she wasn't the only one. Uh, we have we have patients, they were followed over 20 years,
Yeah.
and the very important information that even if the patient believes that everything is fine because 5 years, 6 years has passed, it's never over.
Yeah.
So we have patients who they are very fine for 6, 8 years, and then they relapse and the cancer come back. So this is the best way and the simplest way to reduce the cancer death when patients in remission, even they are tumor free, consuming DDW for 3-4 months every year, every second year, and that way we can prevent the relapse almost with 100%. Those who are coming back and repeating the cure, they it means that they cause a reduction in the DDW, in the deuterium concentration, that challenging all the cancer cells. And if there are small group of cancer cells, that way we can eliminate them, and the patients have a good chance not to relapse in the next couple of years.
Yeah, yeah. So, Gabor, as you rightly mentioned, so right now the 5-year survival rate for lung cancer is around 27 to 30%, right? So, do you think that could increase in next 5-10 years if we have DDW in our therapeutic regime and patients start taking from very early stage if that detected so?
Uh, can you repeat it, please?
Right. So, Gabor, do you think the overall survival rate, especially in lung cancer, where it's very minimal, right? 27%, 30% max. So, do you think with the application of deuterium depleted water, the survival rate for lung cancer, especially in first 5 years, will increase?
So, yeah, I would say that we are not going to replace the deuterium depletion,
Yeah.
the existing therapy. It is very, very important.
Yeah.
We need to integrate deuterium depletion to the existing therapy. 99.9% of the cancer population receiving the conventional therapy. And we have seen very good synergistic effect. For example, combining with DW and, and radiotherapy, we can get a much better final results. We published a paper with a glioblastoma, and those patients who were so lucky that when they received the radiotherapy, at the same time they consumed DW, then the median survival time was 47 months, which is a threefold increase comparing to those who receive only the conventional therapy. And also there are lots of good targeted therapy. So these are targeting those signal system, but the DH ratio also influence and has a big impact. So when we combine the targeted therapy with DW, that even can get a even
Yeah.
much better results.
Right.
So the surgery is very important. If the patient is operable, must operate it and remove the cancer. And after that, much easier to keep the patients in the remission. And I also believe that in the future we can combine DW with different compounds just to increase the efficacy of the deuterium depletion.
Right.
So that was the very first and very important question you addressed, that we need integrate deuterium depletion
Right.
to the existing therapy.
Right, right. And Gabor, as you mentioned about, I know cancer is one application where deuterium depleted water is applicable, and we are seeing a lot of research and so on. Do you think there is also application in other indication area, especially in metabolism or anything else that you can recall?
Yeah, thank you for asking. So one day I was surprised when a patient having cancer and diabetes came back and he said his blood sugar level went down. And I didn't believe it because I was planned to use DW for curing cancer. So
Right.
I went to the Semmelweis University and I asked the colleagues to run a couple of studies with rats to make the rats diabetic. And the question was whether we can reduce the blood sugar level of the rats. And, and it was very important. In the first study, we compared the 25 ppm, which is very low deconcentration, with 150 normal deconcentration. And turned out when the rats received the insulin and consuming the 25 ppm, the blood sugar level was significantly lower. And in the next study, we tested other deconcentration, 125, 105, 75, and it turned out that not the lowest D level was the best. The best deconcentration was between 140 and 125 ppm. And that is a message that the cells can lift up change the D level with 20, 30 ppm. So when I mentioned the sodium hydrogen transport is activated, that may increase up the D ratio with 170 ppm. But regarding the metabolism, we found that the insulin signal system was the best when the D level was lower, below 150, it went down to 135. And then we ran a human study, a phase 2 clinical study, when freshly diagnosed diabetic patients consumed DW with 105 ppm. And within 90 days, 30% of the population, we reduced the insulin resistance. So it's clear that the metabolism also regulated by the DH ratio. And this is the same system, and everything is depends on the, even the metabolism determine the cell growth. If everything is fine and the metabolism is well regulated, then the cells is healthy. Then the mitochondria is damaged, then it goes to the no TCA cycle, and then finally we got the cancer cells.
Right.
So, so I would suggest the people should reduce the D concentration of the body with a 5-10 ppm. The reason that it is said that people should cover the calorie intake mainly with carbohydrate, that means our population leave a much higher D concentration than my grandfather, who ate the bacon, and he was fine, because eating more fat, that reduce the metabolic, the deconcentration of metabolic water, and that reduce a lower deconcentration, and that will help the metabolism to work more properly. That is the signal system which is preferred.
Right. And Gabor, as you mentioned about, it's very important, not just only for the cancer, but there could be a lot of other implication, example for metabolic or diabetes in general. So do we have any way to measure DDW concentration in our body? So is there any way to measure? For example, if patient has a very high volume of, say, DDW over 150 versus 30, 55. So do you have any test or method of detection of DDW in our body?
Yeah. So when we run the study with the 30 diabetic patients, then we took the blood and we measured the deconcentration of the blood. Before the patient started drinking DW, then the blood deuterium level was 140, 47, 150 ppm. When they consumed DW for 90 days with 105 ppm, then the average D level went down 133 ppm. So this is just the opposite, optimal deconcentration for the metabolism. But taking blood is rather complicated. So our concept is now that we have a small device and we precipitate the exhaled water, and we can collect 1 milliliter, and we can measure the water content which is coming out from the lung. And that way we very easily can follow how the deconcentration is changing in the patient. Uh, and it is clear that there is a very strict correlation. The
Oh,
lower the deconcentration of DW, the lower will be the deconcentration of the body. Immediately when somebody stops drinking DW, the deconcentration of the body deconcentration is increasing, just because the normal drinking water is increase up back the deconcentration. So when we will start clinical study, we very easily can follow up the patient's compliance, whether they consume the right water, the right volume, and we can correlate with the efficacy of the DW consumption.
right, right. And Gaurav, as you mentioned, I mean, DDW detection could help, to identify the level of mitochondrial activities, right? A lot of signaling pathway. So do you think there would be also a good business opportunity if someone develop a test that detect DDW in blood, and based on they can recommend something, a preventive treatment, not just for cancer, but a lot of other diseases?
So I would recommend people they should drink it for two reasons. DW.
Mhm.
The one reason is to reduce the incidence of cancer.
Mhm.
For for that uh, purpose, we recommend making a cure every year, every second, every third year, depending on what is the risk factor. So that means for 3 4 months, cause a decrease in the body, and hopefully that can eliminate so group of cells which 4 years later would be diagnosed as a cancer.
Right.
The and repeating this in the last for several years, that could reduce the incidence. And those who just want to increase the health and having a, a better metabolism, we recommend just slightly reduce the D level. So
Yeah.
for these people, we recommend consume DW with 125 ppm or 105 ppm, and they can combine half is the daily water, it is 105, the other is normal water. So in that case, people can drink beer and Coca-Cola,
Right.
but drinking DW with 105 ppm. And
Right.
that would increase the people feel better. They are
Right.
more energized, they can sleep better, and they have more energy.
Right. And as you explained, I think there could be also opportunity for, having as a endpoint in a lot of clinical trials where, the level of DDW concentration, if we can map, we can develop more better drugs for different other indication, as you mentioned.
Yeah,
Yeah.
yeah.
So, Ravi, do you have any question that you would like to ask Gabor?
Yeah, so I think, from a more consumer perspective, how do you see this getting adopted? What are the challenges with someone like you need to address so that there is more adoption?
Mhm. The two challenges is first the science, but I think the science has been solved. So
Right.
many scientists has been involved in the research of DW, and just this year, three big review paper has been published summarizing the research of the last 30 years with DW. But the real challenging is the funding and financing that cancer drug development. And I know that so it is very important to control the drug market to be sure that the quality of the drug is it is what it should be. And it is very also important to educate the society, the oncologist, and everybody who is having key role in treating people. So without the approval, the DW never will be part of the therapy. So it's a only aim is we need the approval. We have to bring it the DW as a registered cancer drug and must integrate into the therapy. And that requires huge amount of fund. Our company, all the revenue what we generated in the last 30 years, we invested back to the research and to drug development.
Hmm.
We are the only company who has a GMP facility to produce DW. So we are ready to produce DW according to the GMP rules, which is essential to go to the clinical study. Uh, and we've got a DW as a drug candidate. Uh, and then we are seeking for partners. We are seeking for pharma companies which has the human uh, sources, how to manage uh, the clinical study, how to manage the production, how to manage all these part of the, which is essential to finally have a drug on the market, or investors who can see that the business potential of the coming out with a new type of drug on the market.
No, I think, it's the passion again comes out with what you say. With the way you've been putting efforts to promote this, funding it yourself, And I think it's just a matter of getting the news out, and then there would be more interest because it is of course you know unlike a large pharma company, it's someone with a lot of passion who's created this whole uh, you know cure to the cancer. I think that's the journey, and I think the little we can do is spread the word. Get you some more people to come and hear you more, and I think that's the start which we look at.
Yeah, thank you. So, yeah, I'm pretty sure that the time will come, but what I don't understand, why should have diabetes for 30 years? So,
Right.
and it's if from a perspective of cancer patients, it's a little bit too long. So, but anyway, yeah, I keep saying I'm working every day, and we try to involve more and more people,
Yes.
and this is the only way we can hopefully will be successful.
Right. And Gabor, just for audience, if you are looking for deuterium depleted water, is your product only available in Europe, or that can be available globally? You can ship anywhere in this world.
Uh, can you repeat me?
Yeah.
If
So we are talking about if somebody is looking for the deuterium depleted water that your company is producing. So is it only available in Europe at the moment, or is it
no,
globally available? We can ship. You can ship anywhere in this world.
no. So today there are several companies worldwide who producing DW and selling, and our products, which is only on the market, is available in Australia, China, Japan, UK, Netherlands. If people order it from Hungary, we are able to deliver it, and which is maybe very important. So the question is how to use DW, and just to help the people, I wrote a book which is called Deuterium Depletion, and it's available on the Amazon. So people n- not only drink the DW, please learn it.
Yeah.
What is the best way to use it? Because in that book, I wrote different protocols depending on the cancer type, depending on the conventional therapy, and I try to share the knowledge of what would be the best way to apply it to get the best results. So even those who are not buying DW from our company, they can get access to the book and to learn how to use it.
Yeah. 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 So I guess now the platform is open for questions. So in audience, if you have any question for Dr. Gabor, you can put it down here and we'll pass it to Dr. Gabor.
Meantime, I can tell you that the European Commission of Research and Executive Agency supporting research with deuterium depleted water. So in Europe, there are universities, Würzburg University, Lusofona University, Oxford, who is working on to test the efficacy and the effect of DW on the brain function, on aging, and metabolic diseases. So we are in the position now that even the European Union is supporting the research with the DW, which is very helpful for us and for me. That it's mean we are getting closer to the final aim. How does incorporation?
Yeah, so we have first question for you.
Ah, yeah, this is a very good question. So the question is, why can deuterium depleted water kill cancer cells?
Yeah.
So the very important fact was, so when I follow the people who consume DW, I always look for why are a couple of people who are not responding so well to the DW. And after a while, it turned out that who were taking lots of antioxidant, then the DW wasn't so effective. So later I told the people, don't take lots of antioxidant. And luckily, Professor Zubarev from the Karolinska Institute later published it, that uh, lower the concentration trigger radicals. And finally, that radicals can trigger the apoptosis and the necrosis of the cancer cells. So the redox balance will not be balanced. And the concept and the lowering D level is so challenging for the cancer cells that will generate the radicals, and in the best case, that finally will trigger the necrosis, and this is how it works. But regarding the DNA and the gene expression is also very important. We published a paper when we followed. The question was whether the deconcentration can modify the gene expression. So we tested over 200 cancer-related genes and over 500 kinase genes. And the cells were kept on deuterium-depleted media and deuterium-enriched media, 300 ppm. And the point was that 99% of the cells responded to the higher deconcentration. So what I said at the beginning, growth hormone bind to the membrane, and that trigger an increase in the DH ratio. So when the DH ratio is increasing, then it will simultaneously turn on hundreds of gene expression, which has a key role to manage the cell growth. So when we suggest people consuming DW, that way we can prevent to activate those genes which are essential to manage the cell growth. So, and this is how we can influence the DNA, and this is how we can uh, get a big impact on the DNA and on the genes.
Right.
Okay, I have a question. Eber, what specific biomarkers or indicators have been used to assess the efficacy of DW in patients?
There are lots of.
Okay.
One of them, and the simplest, is the PSA. So the other male population, the prostate-specific antigen is very strongly correlated with the size of the tumor size. So we have tested lots of people has a higher PSA, they without
Uh,
receiving any therapy. Start drinking DW, we can reduce back the PSA. We we published a paper with the patients who were who didn't accepted uh, the conventional hormone therapy, and for 12 years we could control. And at that time I said, okay, now go and receive the hormone therapy, because DW was good enough to keep the PSA on a low level, and after 12 years he had to go to the hormone therapy, and he's still fine. Uh, for example, there is a patient with a chronic lymphocytic leukemia. So that type of cancer is diagnosed, and he had a huge tumors on the neck and in the abdomen area. But according to the protocol, they waiting that the blood cell number reach the 100,000, and then they receive the chemotherapy. But before that, we had a window, just giving the DW and to see whether we can influence the size of the tumor.
yeah.
So we started drinking DW and disappeared the tumor. And again, we kept it for 10 years. And the reason that the cause effect relationship, when consume DW, the tumor disappeared. Stop drinking after a couple of months, come back. Start again drinking DW, the tumor disappeared. So which clearly shows how it works.
Yeah.
The lower D level triggered the necrosis of the cancer cells, and when no break in the system, then the tumor can come back and
Right.
and grow it again.
Right. And Gabor, does it have similar effect on metastatic cancer, or
Sure.
yeah?
Yeah, the paper we published, it was in breast cancer patients. So in that paper, we evaluated 74 breast cancer patients
Mhm.
who had 135 metastases. So
Oh,
there was patients who had metastasis in two organs. There was some in three or five organs had the metastasis. And the median survival time for these people is about 1-2 years. So in our case, is it goes up about 40 months or more than 4 years or 5 years. But the other, we evaluated the dosage. So in that case, when the patients consume the right deconcentration and the right volume, then 70% we can get a complete regression of the metastasis. So it in the bone, in the liver, in the lung, they disappeared. And that again another uh, uh, evidence that it is important. What is the body weight of the patients? How much they drink? And what is the deconcentration of the DW? If people is drinking not a right volume and not a right deconcentration, then the DW will not so effective.
Yeah. Right. And just in terms of application, Gabor, do you think is any
Yeah, I see it. Yeah. Yeah, we also published a paper when we tested in vitro combining DW with a chemotherapy. And we could prove it that if we combine DW with a chemotherapy, we can reduce the dose of the chemotherapy and can get a good efficacy, but we can reduce the side effect of the chemotherapy because we reduce the toxicity of the chemotherapy. So again, this is another very important issue that uh, how can we manage the cancer patients that using the chemotherapy, not in the right dose, which is used today, and receiving a very, very good response rate. It's very important. The immune therapy, it's I would say quite new, and I couldn't say example that how oh no, sometimes we had. So we have a quite a good response rate even when the immune therapy was used, but we do not have so many patients that I could say that we have published papers and we evaluated hundreds of cases in that case. The surgery, yeah. The other so the other big question is that the surgery. So the best would be if the patients can drink DW for 2-3 months before the surgery.
Mhm.
Always much easier to operate the patients who had consumed DW before the operation. So the tumor is getting a round shape, much easier to remove the tumor and see where is the border of the healthy cells and healthy tissue and the tumor tissue.
Right. And Gabor, I believe there won't be any contraindications for DDW. I mean, there will be no side effect at any concentration, or it may be harmful at below some concentration.
Uh, what was the concentration? Do you mean?
So yeah, so is there any
Yeah.
possibility of getting an adverse effect of DDW use at below some concentration? For example, less than 5 ppm or less than 2. Is there any side effect, adverse effect of DDW so far that we have seen?
Okay. So far we did not see any side effect of the DW. Uh, and if we talk about uh, healthy people, okay, I would say we never recommend patients to drink DW with 25 ppm or 10 ppm.
Uh,
There are uh, other way, others suggest just drink a glass of water with 25 ppm and the rest should be normal water. We recommend people should cover in a, so we do not recommend drinking small volume, very low deconcentration, and big volume, normal deconcentration.
okay.
In that case, when somebody consumed water with 10 ppm or 20 ppm, they reported that they were a little bit drowsy, sleepy, and I think in the future maybe we can find and prove it. A very sharp decrease in deconcentration is not healthy. It is not recommended. So we typically recommend cancer patients start with 105 ppm or 85 ppm.
Okay.
Those who are in the late stage, we recommend start immediately with the 85 ppm. And the other proposal, drinking a given deconcentration within 2-3 months will be the equilibrium. So after drinking the same type of water will not cause any changes in the D level, which means that all the cells is adapting. Even the cancer cells can adapt to the lower D level. So this is the reason we recommend after 2-3 months, change the deconcentration with a 20 ppm lower D level. So after 85, consume 65, 2-3 months, consume 45 or 25 ppm. So this is the way we recommend to go down to the those who had cancer, they feel the consequences of drinking DW because they feel the consequences of a necrotizing tumor. So people can be drowsy, sleepy, a dark urination, can be fever, can be lung cancer starting coughing. And this is because the necrotizing tumor is causing inflammatory inflammation, the reaction, and it became warmer. There are some changes, some pain, and and we say keep drinking, and that will disappear.
Yeah.
So if somebody has a cancer, they feel when they consume DW. Those who are healthy, they feel better.
Yeah. Great.
Okay. I think we are at the end of our questions, and also we are kind of getting close to the time to close this session today. Uh, Thank you so much, Gebor, for giving us such wonderful insights into the research which you've done for more than 30 years. And I think the audience also learned a lot about DDW. Thank you, Wahid, for all the questions which you raised, and I'm sure there would be more, questions as we go on. And I think the entire conversation gets posted on our LinkedIn channel, and we will get more requests coming in, and we will definitely reach back to you, Gabor, with anything which we like to hear back on that. So, with that note, I would like to end the session. In case you want to speak something, Gabor, welcome.
Yeah,
Uh,
I would like to say thank you. So
yeah.
that's it's a great help for me and for I hope everybody, if we can has a surface to share the knowledge and thank you again. So keep working on it. I am ready anytime if you feel it's useful, and if you have a progress, we are happy to share the new information and the new knowledge.
Okay, absolutely brilliant. And to the audience, please do join us on our LinkedIn page where we keep announcing, our Tech-novation events, and you would obviously get a lot of insights around deep tech and the markets which we track. And thank you once again, and everyone have a good evening and a good night wherever you are. See you
Thank
again.
you.
Cheers. Bye bye.
Thank you. Thank
Bye
you.
bye.